Hi, Eva.
Hi Gordon, how are you?
I'm very well. So we've just been talking downstairs about the advances in materials like graphene and biomaterials and looking at some of our fabrication aspects. Of course, another important part of our work is the integration of living cells into our printing protocols. Can you tell us something about how we prepare those cells for printing in a facility like this?
So those cells that we have, we culture these cells in this lab. It is a PC2 facility. So we have a risk group rating, which allows us to work in an environment that the cells that as a scientist, we can be protected from those cells, but the cells can actually be handled in a very safe manner. So we have cells which we obtain from human patients, cells that are from immortalized cell lines, cells that are from various tissues that we might be interested in expanding and differentiating down certain lineages. So that's where these cells are stored in liquid nitrogen. Here we have a couple of examples of where we store our liquid nitrogen to try and ensure that they're maintained at a very cold, in a very cold environment that allows them to be able to be expanded, taken out of the storage and expanded over a long time. We also have an environment where we try to nurture those cells in terms of providing the right growth conditions to expand. So we have incubators that we just go around. So we have here incubators that provides a warm 37-degree environment, that has air that is good for maintaining a pH. It's an environment that's good for the cells to grow and be conducive to cell growth. So.
Right. And so do you monitor the health of the cells during these processes, or do we do that afterwards?
So quality control of our cells is very important. So knowing that a cell that is derived from a skin biopsy is exactly that in the first instance is very important. If those cells are then reverted to their pluripotent cell state, a stem cell state, we have to identify that those cells do maintain the ability to be pluripotent. So yes, absolutely.
And how do you test that?
So there are a number of markers that we use, proteins that are expressed that are reflective of that pluripotent stem cell state. But we also, more simply, take them out of the incubator when they're grown in the tissue culture plates, put them under the microscope.
Let's go and have a look at the
Put them under the microscope. We have Sarah there. She's looking at some of our cells. Put them under the microscope and ensure they actually have the phenotype that we expect to see.
So under the microscope, is it mostly you're looking at the shape of the cell, or are you actually scanning it to see if there are those markers we just spoke about?
Yeah. So there are different levels of characterization. There's the level of actual just basic morphology, that high nucleus to cytoplasm ratio that is characteristic of, say, a pluripotent stem cell. But when it comes to looking at those markers, one of the simple methods that we use is immunofluorescent cytochemistry. So we use an antibody that recognises that specific target protein, and if that antibody is linked to a fluorophore, we can see that under the
glows in the dark.
Glows in the dark. We see that under the microscope. We can add a lamp that will mercury lamp that will excite that fluorophore, and we can see it glowing in the dark. Exactly.
Right. So we get the cells in, we store them, we put them in an environment that they like to stay in, and we characterise them all along the way.
And we grow them. We put them in the incubator to expand, to scale up the number of cells that we need. When we go through the process of bioprinting, we want to create a three-dimensional tissue, not a few cells in there. We want to actually have a high cell to biomaterial mass. And so what we want to do is scale up the number of cells that we have to be able to encapsulate into the biomaterial, which we will do next door. But that process of encapsulation, of working with the cells, it's very important that we do it in a safe environment such as we do in the biological safety cabinets that we have over there.
So are there other characterization tools for the cells that you use in here, or should we go have a look at the bioplotter?
We can, it's a process of going back and forth. Right. So whilst we scale up the cells here in the first instance, we go to use a bioplotter and we print those cells in a structural form that we'd like to have, but then we bring it back into this lab and we continue the culture and growth. The characterization also extends to, say, a functional analysis of, say, if you're looking at nervous tissue or neural tissue, you'd be looking at multi-electrode array, so look at those electrophysiological responses, or you'd perhaps be using confocal microscopy to look at calcium flux, how those cells network and show coordinated flux.
Okay, well, let's go and see how we build all those cells into the actual 3D printed structure. So having prepared the cells in this sort of environment, we go take them to the bioplotter, and then create that structure.
Yeah.
But of course, we'd want to bring that structure back into this environment to interrogate the cells again, see if they're still living, if they're proliferating, and maybe even turning into the types of cells that we want them to. Is that correct?
That's correct. Another really very important point is actually demonstrating that the biomaterial that we're actually creating, that nested material, that encapsulated material, is supportive to just basic cell viability. So we have cytocompatibility testing that we also do. That's another aspect.
So if you were to bring the cells here, what would you actually do? You'd load them into this 3D bioplotter?
So
maybe if you just point out how we do that?
Certainly. So what we have is a 3D bioplotter here. There are cartridges that we load our biomaterial that has the cells encapsulated, or just the biomaterial alone. We don't need to have the cells necessarily at this stage encapsulated. We bring that already prepared in a cartridge. That cartridge, almost like a syringe cartridge, that cartridge is loaded onto these cartridge holders on the right-hand side. They are temperature controlled, so it's conducive to bioprinting at particular temperatures that allows the form of the filaments to be preserved.
So the cell, at this stage, the cells would be added to the bioink formulation, is that correct?
There are two ways we can approach this. We can actually allow the cells to be encapsulated into biomaterial, or we can actually seed afterwards. So this is the part where there's an option of one or the other. Having said that, the biomaterial that we bring, that we have prepared under sterile aseptic conditions in the tissue cabinets we have over there, that is then brought here. And yet again, we are in an aseptic environment. So we have a BSc class 2B cabinet that we introduce ourselves, or our biomaterial with ourselves, into this environment.
Right. And I know recently we've been successful in printing structures containing induced pluripotent stem cells. That's an amazing advance, isn't it, for both fundamental studies but also potentially for clinical applications?
Absolutely. Pluripotent stem cells have the ability to differentiate down all the different lineages of the body. So we can allow the cells, it's what's quite remarkable is that the cells, which are very difficult to handle in native culture, we can actually incorporate that into the bioink, which allows the viability of the cells to remain viable, and to promote the incorporation of the cells into the material. So yes, we can print the material under with the bioplotter into a defined structure, a three-dimensional structure, and then that material, once we take it back into the PC2 lab, we can culture in a media with the growth hormones and other supportive media additions to the media that allow the cells to differentiate down different lineages.
Right.
So it's quite exciting to be able to create these very high density structures that the cells are able to form a specific 3D tissue.
Yeah, and so that ability to create those structures gives us fundamental insights into how things behave in 3 dimensions.
Yes.
And obviously what we're discovering is it's very different from 2 dimensions.
Yes.
And
We're not a flat body.
Yeah.
We're not a flat surface. Our cells in the native environment in our bodies interact with other cells in that 3-dimensional space, interact with the extracellular matrix, and that biomaterial provides that extracellular matrix that the cells can have support to grow further and to really integrate into the 3-dimensional tissue.
So it must really bring into question the validity of pursuing studies in 2 dimensions in terms of conventional cell culture.
Yeah, it's a growing. It's been a growing concept that 2D is great for fundamental initial studies, but taking these studies, from whether it's the cancer cell biology or basic tissue biology, taking cells from a 2D matrix, a 2D monoculture to a 3D matrix is essential to understand tissue biology.
Great. Well, thanks for giving us that insight into the preparation of the cells for 3D bioprinting and then how they continue to be interrogated after 3D bioprinting in order to understand how they develop in 3D and how we can manipulate their development in 3 dimensions. Great stuff.
Thanks,
Thank you.
Gordon. Thanks for your interest.
Thank you.